Hi everyone, you are watching Blackboard and welcome to our lab practical series. In this video, we'll be discussing an easier way to streak a Petri plate. Streaking is an important step for accurate diagnosis of microbes as isolating separate colonies is quite a task. But not anymore. Here is how you can streak a plate efficiently and easily to obtain isolated colonies. I'll show you by animation and on the real Petri plate as well. Put a drop of the sample on side. Make a well by rotating the wire loop. Make 3 lines by touching the well. This is called primary streaking. Next, make 3 more lines by touching the primary streaks. This is called secondary streaking. Repeat the same procedure for tertiary streaking as well. At last, make a tail without touching the tertiary streaks. And this technique is called quadrant streaking. A good streaked plate should only have 80% of its area covered. Make sure not to touch the sides of the plate with wire loop to avoid contamination and not to overload the plate. Now, here is the real plate. Label it according to your experimental needs. Red hoard the wire loop to kill all the microbes if present on it. Take sample. Put a drop. Make a well. Then primary streaking, secondary streaking, tertiary streaking, and here you go with the final tail formation. Cover the plate and red hot the wire loop again before putting it away. Now, if you have any questions regarding this procedure or you want to know how media is prepared, please mention in the comment section below. And if you found this video helpful, like the video and subscribe my channel. Thank you.